In this video, we'll have a look at some of the basic ways things get across the cell membrane. So we're going to talk about membrane transport. Now just remember, one of the key things that a cell has to do is really carefully regulate what leaves and enters the cell. And based on the chemistry, the size of the objects to come or go, there are different techniques that are used or different procedures. So we're going to have a look at some of the basic ones. So let's remember that the plasma membrane is selectively permeable. So it allows some things to pass through. You know, some things can just rapidly cross cell membranes because of their chemistry. And we could break all the mechanisms into two big groups. Does it use energy, or is it able to do it without any energy? And we'll look at the passive mechanisms first. We'll just mention those. Those don't require the use of ATP. And this is really driven by random molecular motion of particles. Now, which ones fall into this category? Things like filtration, diffusion, and osmosis are classic examples of the passive mechanisms. Not surprisingly, if we have passive mechanisms, we also have active mechanisms. And the hallmark of those is that they do use ATP. And collectively, they tend to be called active transport mechanisms. And another example is vesicular transport. Now, we also have carrier-mediated mechanisms, which use a membrane protein to move things across. So these are just some of our basic subgroups. We have basically our passive versus active. Under passive, we have filtration, diffusion, osmosis. Under active, we have active transport, vesicular transport, and also we have another type of transport which is called carrier-mediated. It's good to get familiar with sort of where these terms fit in to the overall structure, because we're going to look at each one of these in detail. Now, filtration is basically particles that are driven through the membrane by a physical pressure. And filtration happens, typically we see it happening in things like capillary walls. That's one of the greatest examples. And we're talking about very small things, water, small solutes, and they fit through the gaps of the capillary walls. They're small little gaps that some things can get through. Now this is a good mechanism because, of course, it doesn't depend upon ATP. It's energy independent. But it allows the delivery of waste and nutrients to the tissues. You know, one example of that would be in the kidneys, the capillaries in the kidneys and how they function. And we'll look at that next semester in detail. Here we can just see it in action, that here we have the individual cells that make up the capillary wall, and you can see that there are some gaps between them. And here on cross-section, you can see one gap right there, and how, if you're the right size, right chemistry, you can enter the tissue around the blood vessel, or you can enter the blood vessel itself. Now, we could also look at simple diffusion. Simple diffusion is the net movement of particles from a place of high concentration, not surprisingly, to a place of low concentration. Here I don't need any energy added to the system because this is due to basically spontaneous molecular motion. And it's kind of, I always think of, if you've ever been to amusement park and you see those bumper cars, it's kind of like that, is that the molecules collide and bounce off each other. Now, not surprisingly, with simple diffusion, things like to go from areas of high concentration to areas of low concentration. They diffuse in that direction, from high concentration to low concentration. And of course, we call that diffusing down their concentration gradient. We don't need a membrane to get across. This is just a simple type of diffusion. And we're not talking necessarily across membranes. But if there is a membrane and it has the right chemistry, it could diffuse through a membrane if it's permeable to it. Now, there's a whole number of things that affect whether you're going to undergo diffusion and how much and how effective your diffusion is going to be. One is temperature. Not surprisingly, temperature, if I increase, this should be increase temperature, then I increase the motion of particles. They're going to move more, put more energy in, particles move more, they're going to diffuse further. The next thing is molecular weight. Not surprisingly, the very small molecules will travel farther faster, whereas the larger ones are slower to move. So larger ones are slower to move. Also, what is the difference, concentration gradient? You know, if I have two concentration gradients that are almost identical, well, I'm going to get less stuff moving from area high to low if that difference is very, very little. So we call it the steepness of the concentration gradient. If I have a big difference, an increase in difference of concentration gradient, then I'll have an increase in the rate. The next one is the membrane surface area. Not surprisingly, if I'm talking about diffusion across a membrane, the higher or the greater the surface area, the faster the rate. There's just more opportunity. There's more surface area for this to occur. And also, how permeable is the membrane? And not surprising, once again, that if I have increased permeability, I'm going to have increased rate of diffusion. Now, we'll talk a little bit about osmosis. Now, osmosis is, let's start actually by defining it. Osmosis, it's the net flow of water through a selectively permeable membrane. And what does that mean? Well, basically, when we're talking about that, we're basically saying water moves, so it's about water movement, from the side where it is more concentrated to the side where it is less concentrated. Okay? We're really talking about solutions here. Now, why are we talking about the movement of water? Is because this is a case where the solutes in the water cannot pass through a membrane. But what happens is if I had an increase of solutes on one side of the membrane versus the other, the side with a higher number of solutes will draw, almost like a sponge, suck the water through the membrane. So solute particles that cannot pass through the membrane draw the water from the other side. We see this all the time, when we're looking at osmosis, and we'll talk about some special cases. Now, water can diffuse through a phospholipid bilayer, but often when we're looking at an actual membrane, that there's these channels called aquaporins, aqua for water, and these are channel proteins in the membrane. They're specialized for water passage. And the thing that we should also know is that it's not stagnant, the number of these aquaporins. These aquaporins in many places of the body can be increased in number or decreased in number to increase the amount of water flow. Now here's sort of the classical sort of showing of what osmosis is. Here we have a semi-permeable membrane running down the center. Over here, all these little dots are water molecules. Over on this side, we have a solution. So these are not water molecules, these are something in the solution. And of course the bluey ones are the water molecules. So what happens with osmosis is these particles cannot diffuse through. They just can't, because their chemistry, their size, or whatever, they can't diffuse. So what happens instead is, since they cannot move, what happens is water will move to try and even out the solution. And that's what we see happening, is that we'll have a net movement of water across the semi-permeable membrane, and it's almost like it's trying to dilute this solution that would I'd end up with, theoretically equal amounts of solute on each side. It's not going to happen in this case because remember the solutes cannot cross. So water is going to leave. Water is going to leave this side and enter this side. So this area is going to have a loss of water. The net movement of water is over to this side. Now there is something that counteracts it. Like you would think, okay, will this go forever? Will all the water move over here? Well, no. In most circumstances it won't. Because here's one thing that happens. There's osmotic pressure. Osmotic pressure is basically the pressure caused by this flow of water in. I also have hydrostatic, hydro for water. Think about this. As more water is coming over to this side, this column of water is getting bigger. And that represents a weight or a force. And what happens is, it's kind of like water behind a dam. Is that, if I have more water behind a dam and I open the gates and the water's going to come through it faster, is that this hydrostatic pressure is going to build up and it's going to now try and force water back this way. So now I have two forces at work. I have basically the osmotic pressure or the osmotic force, which is bringing water over here, and water's building up here. But the fact that water is building up here, basically because water has weight, etc., it's going to have its own pressure, and the hydrostatic pressure is going to start to force water back. And eventually I should hit a point of equilibrium where the flow of water in this direction is equal to the flow of water in that direction. So once again, the two forces in this case were the osmotic pressure and the hydrostatic pressure. And remember, one of the key features of this is that we're talking about flow of water. It's not the solute, it's the flow of the water through a semi-permeable membrane. Now, we can put some definitions to a couple of those terms. Osmotic pressure is the hydrostatic pressure required to stop osmosis. That's one way you can define it. Now, also there's reverse osmosis. I'm not going to spend much time on that, but it is an industrial process. It's used in medicine and certainly things like dealing with kidney disease, etc., and that where they can draw off some of the toxins from the blood. But so there's reverse osmosis. It's basically applying a mechanical pressure to override the osmotic pressure. We really don't see that in the body in any great way, and it's really the osmotic pressure that we're talking about. Now we're going to stop there, and when we come back next one, we're going to talk more about osmolarity, tonicity, isotonic, hypertonic, hypotonic.